In 2011, a DNA lab opened a cold case that was 3,500 years old. The suspects weren't in police files. They were alive, walking around, living their lives as Cherokee citizens. For 2 centuries, Western historians treated Cherokee oral tradition as folklore, stories, not evidence. Then geneticists found something locked inside Cherokee mitochondrial DNA, a marker called haplogroup X2A, carried by a small fraction of Native Americans, that traces back over 15,000 years. What it proved didn't just back up what the Cherokee had been saying all along. It forced scientists to rethink the entire timeline of how North America was first populated. But here's where it gets messy. The same evidence that vindicated Cherokee oral history also got hijacked by a theory you've probably heard floating around online. One that has nothing to do with what the DNA actually says. Today, we separate the real science from the myth riding on its back. For most of recorded history, the origins of the Cherokee seemed beyond debate. They were a mountain people. The high valleys of western North Carolina, eastern Tennessee, and northern Georgia had been their homeland since before memory began. Their sacred sites, their language names for rivers and ridgelines, their oral traditions, all of it rooted here in the Blue Ridge. That story was comfortable, and it held until linguists looked at the language itself. Cherokee is an Iroquoian language. Every other member of the Iroquoian family, the Mohawk, the Seneca, the Onondaga, the Tuscarora, lived more than 800 miles to the north and east. Great Lakes country, upstate New York, the St. Lawrence Valley. Not one other Iroquoian language was ever spoken this far south. By the 19th century, researchers were already asking the obvious question: how did an Iroquoian-speaking people end up in Appalachia? Running divergence models on core vocabulary and sound shifts, linguists estimated the Cherokee language split from its northern relatives roughly 3,500 years ago. That placed the separation in the late Archaic period, long before Mississippian civilization, long before European contact, long before any written record. Something moved south, something significant, but archaeology could show you pottery styles and settlement patterns. It couldn't tell you who moved, how many, or what they left behind in their cells. That required a different kind of evidence entirely. When geneticists began mapping Native American ancestry in the early 1990s, the framework they constructed was almost elegant in its simplicity. Nearly 97% of all Native American maternal lineages fit into 4 founding haplogroups, labeled A, B, C, and D. Each one traced back to East Asian source populations, each one consistent with a Beringian land bridge crossing during the last ice age, roughly 15,000 to 20,000 years ago. The model matched the archaeological record. Papers were published. The debate appeared settled. Then, the remaining 3% started causing problems. A 5th lineage kept appearing, specifically concentrated in one region of the continent. Haplogroup X. And unlike the other 4, Haplogroup X did not have clear East Asian parallels. Its closest genetic relatives were found in Europe, in the Middle East, in populations that had no obvious connection to Beringia. Every few years, someone uses Haplogroup X to argue that Native Americans are secretly descended from Europeans, or that ancient Israelites reached the Americas. None of that is what the data shows. The Native American variant, designated X2A, diverged from its Eurasian relatives at least 15,000 years ago. It crossed into the Americas with the founding population. It is not colonial contamination. It is not proof of ancient contact with the Old World. What it is a geographic fingerprint. And once researchers figured out where that fingerprint was concentrated, everything changed. In 1999, 2 independent research teams, Michael Brown at Emory University and David Glenn Smith at the University of California, Davis, published simultaneous findings confirming that X2A was real, ancient, and unevenly distributed across North America. 3 years later, researcher Ripan Malhi assembled the most comprehensive mitochondrial DNA dataset anyone had attempted at continental scale. 1,612 samples from dozens of native North American populations. When the team ran spatial analysis and mapped haplogroup frequencies against geography, the continent organized itself into clear clusters. Haplogroup X concentrated heavily around the Great Lakes, not the Southeast, not Appalachia. The Great Lakes, precisely the region where every other Iroquoian language group had lived before European contact. Now overlay a second map, the pre-contact distribution of Iroquoian languages. The spatial overlap is not approximate, it is precise. Now add a third layer, Cherokee oral tradition. The Kituwa stories, passed through generations of elders, describe a migration from the north, a journey made long before it could be written down, mapped, or sequenced. 3 completely independent lines of evidence: genetics, linguistics, indigenous memory, all pointing to the same origin, all pointing to the same direction of movement. When that happens in a cold case, investigators have a word for it: corroboration. But here is where the case nearly went cold permanently. The primary evidence was almost destroyed. In the winter of 1838, the United States government forcibly removed approximately 16,000 Cherokee people from their ancestral homelands and marched them west, through ice, through starvation, through disease. An estimated 4,000 of them died before reaching what the government called Indian Territory, present-day Oklahoma. 1 in 4 people gone. In population genetics, what follows an event like that carries a specific name: a bottleneck. When a large portion of a population is suddenly removed, the genetic profile of the survivors no longer reflects the full diversity of the original group. Rare lineages vanish, not because they were less significant, but because their carriers happened to die on a winter road. Common lineages become overrepresented. The signal gets compressed, distorted, incomplete. Every DNA study of modern Oklahoma Cherokee is, in part, a study of which genetic lines survived a forced march. What was lost may never be fully reconstructed, but the case was not closed, because not all of the Cherokee were removed. A small number fled deep into the mountains of western North Carolina, into the hollows and ridges the removal forces couldn't fully reach. Their descendants are the eastern band of Cherokee Indians, still living in the original homeland, still in the mountains where the Kituwa stories were first told. 2 Cherokee nations, separated by the same catastrophe, each one carrying a slightly different piece of the genetic record. The eastern band offers researchers a window that wasn't filtered through the 1838 bottleneck in the same way. Comparing lineage frequencies between both populations, scientists can begin to reconstruct what Cherokee genetic diversity looked like before the destruction, and sharpen the picture of the migration that started 3,500 years earlier. The work is still in progress, but it is being done differently than it once was. The technology to fully resolve this migration exists right now. Ancient DNA extraction can recover usable sequences from remains thousands of years old. Statistical models can distinguish a single, large population movement from centuries of gradual intermarriage. The answer to what actually happened in the late Archaic period, who moved south, what they displaced, what they left behind, is scientifically within reach. But the Cherokee are not subjects of this investigation. They are directing it. The Cherokee Nation maintains strict protocols governing any genetic research involving their citizens or their ancestors. Data sovereignty belongs to the community. Research requires genuine tribal partnership, not permission forms signed and forgotten. Several ancient DNA collaborations are reportedly in development under exactly those terms, designed to answer the questions that mitochondrial data alone cannot close. Science spent decades taking without asking. The next chapter will be written differently. In 2011, a laboratory in Washington state opened a cold case 3,500 years old. In the 19th century, linguists had found the first clue: a language with no business being in Appalachia. Long before either of them, Cherokee elders already knew. The Kituwa stories had carried the memory of a northern origin across every war, every forced removal, every generation that was told their history didn't matter. Western science spent 2 centuries developing the tools to confirm what Cherokee oral tradition had preserved all along. The cold case was never really cold. The evidence was always there, inside the people who survived.